Thank you so much, Patrick, and hello, everyone, and welcome. I'm happy to introduce us to our April iteration of our Catalyze webinar series. We are very lucky to have today's presenter, who is one of our very own technical project leads for the Catalyze program, Dr. Carmella Moody. Carmella is going to guide us through a particularly complex topic today, which is regulatory submissions, and in particular, the essential data and documentation required for successful CMC submissions. Carmella brings a wealth of knowledge and experience in regulatory affairs, as well as the development of small molecules to today's discussion, which will cover drug substance and product characterization, manufacturing processes, analytical methods, and stability data. As we said, time will be reserved for question and answer, at which time Carmella will be joined by Dr. Mark Levy, who is another Catalyze technical project lead with 20 years experience in medicinal chemistry, farm tox, and CMC. So very much looking forward to that discussion. I'll also note before we begin that we have several members of Catalyze leadership on today's call. From the NHLBI side, we have Mike Peck, Rahul Thacker, and Nitin Agarwal. And from the side of the Catalyze Coordinating Center, Diana Severin-Stevens and Sudhi Roshant. So please feel free to engage with us in either the chat or the Q&A box, and we'll be sure to be responsive. So at this time, Carmella, I'm happy to turn it over to you. Oh, Carmella, you're on mute. Sorry.
Now, thanks for joining us today. I'm looking forward to going through this and talking to you a little bit more about information specifically that goes in the different sections of the CMC section, which is now Module 3 of the Common Technical Dossier or document. So next slide. I wanted to bring this up again. I thought this was important enough that I wanted to highlight that. Please, if you have any questions that we don't answer, or if we can't get to every question today, please send us an email at catalyze_info@rti.org. And we'll try to make sure that the right person responds to you. Our goal is to be teachers and helpers for the Catalyze program. So please use this if you have any further questions, which I hope you will. Next slide. I also wanted to provide you up front, instead of at the end of the presentation, some critical references for CMC or Module 3 for INDs. The first two, the IND content and format for phase 1, and then INDs for phase 2 and phase 3 CMC, those are really old. They were back in the early 2000s, but they're very relevant today for the baseline information that you need to have for phase 1 and then phase 2 and 3 INDs. I think it's important for you to review those because we have a lot of creep of information that people think needs to go into a submission. And I think if you review those carefully, you'll be surprised at the minimum amount of information that's needed. I did provide the link here for gene therapy. We're not going to talk about gene therapy. That's one of the future presentations I hope that we'll be able to have. And then there's another guidance for industry. It's a guidance for industry, the M4Q. And the CTD quality August 2001. And mainly I included this so that you would be able to know the headings that are in the quality or CMC section for an IND and the common technical document format. I want you to note, though, that the guidance information that is in there is mainly for NDA or BLA, okay? So it's a lot more information indicated that's needed than it's actually needed for early phase. So keep that in mind when you're reviewing it. Review that really for the names, the numbering and the names of the headings. And then the general search for the FDA guidance documents. You can click on that and put it, and you type in your subject, and you can find the documents that you need. Next slide. So I'm sure most of you have heard that the format for the day is the common technical document, and that all documents or all regulatory submissions that are considered commercial INDs, which are INDs for products that will eventually be marketed. So it includes most of them except for a few, must be submitted to the FDA electronically. And if you need help, RTI can help you with electronic submissions. And then all electronic submissions must be in the common technical document format. So not the 1 through 9 sections that you will find or that you are used to using. It's in the specific common technical document format. And module 3 is the quality or where the CMC data is included. If you have any questions about ECT or eCTD or CTD format, let us know and we'll try to help you. Next slide. So an overview of the presentation. There was no way that I could cover everything that I wanted to cover for drug substances and drug products in an hour. What I did do is I picked the sections that I thought needed a bit more explanation or information to be included. And the other thing that you're being introduced to is the common technical document format and the granularity of the drug substance and drug product sections for module 3. There are some specific things you need to learn if you don't already know this, is that there are specific numbered headings and the actual headings that are defined. And ultimately, when you put together your document, each numbered section ends up being a PDF file that's submitted electronically. And important point is note that the numbering scheme and the heading names must be followed and you cannot change them. You can change the information underneath or add a heading underneath, but the ones that are provided here and in that document I provided you are set and are the standard for the US as well as other countries. Next slide. Okay, so we're going to talk drug substance. Next slide. So this is the information, the actual sections of the drug substance section, showing you the granularity of the files that end up going into the drug substance section. The information that's in bolded, those are the ones that I'm going to go in a little bit more information. And the ones that are grayed out are actually the sections that are not used for early phase or phase 1 submissions. And again, this is just to help you understand the granularity. Next slide. So first we're going to talk about structure. The 32S12 structure, the information that's supposed to go in there is evidence to support the chemical structure or the biological structure, if it's a small molecule or a large molecule. And this is really important. I think this is information that's oftentimes overlooked as being important for inclusion in an IND. But you need to know things about absolute stereochemistry, molecular formula, mass spectrum, and then you need to provide information on amino acid sequence and glycosylation sites, molecular mass, things like that. So basic information to let the FDA know that you know the structure and information about your molecule. Next slide. So then we go into general properties. And again, it's important. I think there's sometimes there isn't an understanding that the FDA expects you to be able to provide some general property information to qualify your molecule. And so things like color, melting point, optical rotation, partition coefficient, and then for large molecules, information on biological activity and proposed mechanism of action. So this is really important. You have to include this. The first two sections, the structure, excuse me, general properties are the first two sections that let FDA know you know what you're talking about. Next slide. So then we're going to go into the drug substance manufacturing and see that we've changed from S1 to S2 in the numbering. The description of the manufacturing process and process controls is important. Usually you include, or all the time you include, a flow chart describing the manufacturing process, and then you provide a narrative description of each step in the manufacturing process following the flow chart. And you cover things like critical steps, equipment used, and process controls. But for phase one, the information can be very general. And things like the equipment are defined by function. You have descriptive process controls, and you include things like typical process details such as time and temperature, but you don't want to include very tight information, exact, stir for 10 minutes. You want to say typically stir for around 10 minutes. Things that don't hook you into specific information in your manufacturing process. And so this is a good example of an early phase flow chart for biologics. And I forgot to say earlier, just a minute. One of the questions that, or multiple people ask questions about what's the difference between the information that's needed for phase one and phase 2 and 3. So what I did is I tried to include very, very general baseline information for the different sections for phase 2 and phase 3 on the slides. I'm not going to specifically cover that because we don't have time to go through all that. But again, when you go back and you look at the PDFs or you look at the presentation, review that information. It's very high level information to help you get oriented for just differences between phase 1 and phase 2 and phase 3. Next slide. So control of materials. This is really important. The materials that are used in the manufacture of the drug substance, all of the raw materials, starting materials, the solvents, reagents, catalysts, all should be listed in a table and identified to where these materials are used in your process. And for biotech, you want to list things like your monoclonal antibodies, your enzymes, and this is where you provide information on your cell banking system. And what you also want to do is you want to have a column that addresses the quality of the raw material, such as USP or NF or whatever. I'll talk a little bit more about materials of animal origin later on, but my suggestion to you is if you could avoid materials of animal origin, do. And at a minimum, if you're using materials of animal origin, you must have a BSE TSC certification. Next slide. So it's important. One of the other things that I like to talk about, or I think it's important to talk about for an IND, is the impurities. And so impurities is anything that is not the product. So it's really important that you understand you have inorganic impurities that come from your reagents and heavy metals, things like that. You have organic impurities like your starting materials or your degradation products. And then you have residual solvents. And it's important that you have an understanding of each one of those impurities in your drug substance. So section 3.2.S.3.2 impurities is not necessarily one of the sections that you need to have a lot of information in for phase 1, because you address your impurities in your specifications with appropriate limits to assure safety. And one of the other things that's important to understand is it's important to understand your impurities, because what you want to do too is you want to make sure that those impurities, which are the ones that you show up in your batch manufacturing process, you really can't get out or end up at higher levels. You want those to be in your toxicology batches so that you can get tox coverage of those impurities. And if you have any questions about that, let me know. But it's important that your tox batches oftentimes don't need to be as clean as your GMP batches. Next slide. So here we're talking about the specification for the drug substance. And basically, your specifications are a list of tests with appropriate parameters for that must be met that are used for release and stability testing. And for phase 1, and also for other phases, for 2 and 3, you define the critical quality attributes for your manufactured drug substance, which are really the characteristics that should be maintained within a limit or a range to make sure that you have the desired product quality. And you propose acceptable limits for those critical quality attributes. And you should provide chromatograms or things to support the information that you're providing, the data you're providing to support your specifications. And another important thing is that to identify your impurities, oftentimes you do this by a stability indicating assay that is usually an HPLC assay. And it's important your impurities can be identified by relative retention time or an appropriate measure. At phase 1, you do not need to know the actual structure or the name of your impurity. Next slide. And this is just an example of small molecule specification. You see that you have a battery of tests that you use for release of your batch. And then for stability testing, you choose a subset or all of the same release tests for your stability study. You choose the ones that are actually going to change. I think that's all for this slide. Next slide. So the other thing you need to have is you need to provide the FDA with your analytical procedures that you use to qualify your specifications. And the information at phase 1 is provide a high-level method descriptions. You do not have to provide very specific information, and you do not have to provide validation data. One of the things is, I stress this, is that your validated analytical methods are not required at phase 1, but they are at phase 2. But in my experience from lots of years of doing this, the sooner you qualify your methods and you get a good stability indicating method in place, the better off you are, because again, your analytical data is your currency for the FDA. And so that's just my little work. You don't have to follow it, but that's the scoop. Next slide. batch analysis. So you are going to provide information on each batch of material that starting at your GLP studies and carrying through all of the batches that are used in your clinical studies. You want to make sure you include the lot number, the manufacturer, the manufacturing site, and the date of manufacture of the drug substance. And also too, you probably want to include your route designation of the drug substance if that changes. And another bit of information to include is really, you can put the number of the study, so the GLP study, or information about the clinical study where those batches were used. And again, you want to provide your batch analysis starting at your GLP tox studies and carried through for phase 1 and future batches. You want to include a C of A for the batch of clinical trial material in your IND. Now, there was a question that's, that was, do you have to manufacture your batch of clinical trial material before the IND? And the answer is you should probably have your drug substance batch manufactured, because you're going to have information on your drug product batch for the IND also. So you go ahead and manufacture your drug substance batch or batches before you file the IND and have that information in the IND. We'll go over this a little bit more later, but for your drug product, you can wait. You don't specifically have to have your batch analysis data or your certificate of analysis for your drug product batch in the IND, but you should have negotiated with the FDA at your pre-IND meeting that you're going to provide it before you start your clinical study. Next slide. So justification of specification. So it's important to understand why you chose your critical quality attribute and why you provided the specification, or you limited the specification to the range that you did. For phase 1, the specific focus should be on critical safety issues. The information can be fairly general, but you want to provide, well, ICH Q6B provides guidance on the general principles for setting and justifying specifications. But if you have trouble with the kind of information you need for early phase, let us know and we'll help you. Next slide. Okay, reference standard or materials. Usually for your early phase, you select a batch of your drug substance for your working standard, and you test it just like you would for release, but you generally add additional tests for identity and purity beyond the standard for batch release. And then you use that batch as your reference standard for all future clinical trial batches that you manufacture. And as your development progresses, you're going to fully characterize and set aside material that becomes your primary reference standard that is compared back to your working standard. Because your reference standard is gold, and it's especially true for biologics because it's what you compare all of your other batches, your future batches to. So you need to consider how much of that you have and treat it like gold. And then your reference material, it generally should be of the highest purity possible. Next slide. I put some information about container closure system. The FDA considers it it's the sum total of the packaging components that protect the drug substance. So you should include a brief description of the components, a description of the assembled packaging system, and any precautions needed to ensure the protection and preservation of the drug substance, like whether you're using desiccants or anything like that. There's a good older document for container closure systems for drugs and biologics. The information is here. It's good to know, and it's important to know. Next slide. It's important to know that your stability data is in the specific container closure you chose. You don't provide FDA with stability data from just, how you stored it on the bench. The stability data they want is your drug product substance in the actual container closure, or a mock-up of the container closure that you actually use for your drug substance. In this section, it's not necessary to provide a stability protocol, but I think most people do. And here you provide conclusions from the stability study that you've performed so far. And usually that information is to let you know when it's time to reset your retest time points. Next slide. So the stability data goes in a different section, and it should be included in tabular format. Again, you include the lot number, the manufacturer, and all the information that you actually included for the batch analysis. And that information allows you and the FDA to understand your overall stability program and how you're keeping that material safe until that drug substance is incorporated into drug product. Next slide. Okay, so now we're going to go to drug product. Okay, this is the granularity information for the drug product. Again, the things that are bolded, I'm going to go through and talk a little bit more about. The things that are not bolded or grayed out are the ones that you don't usually include for early phase. Next slide. Description and composition of the drug product. And what's important here is a table listing all the components used in the manufacture of the drug product. So you want to include all the components that appear in the drug product and any components that actually were used up in the manufacturing process too. So for phase one, it's a tabular format, and you put a table or a column to include the quality of the inactive ingredient, whatever it is. Next slide. The batch formula, again, that's the quantitative information for your batch. For all of the components, whether or not the component appears in the final batch, and it's specifically for a batch, for 10 kilograms or 100 kilograms, whatever it is. Next slide. You, again, like the drug substance, you provide a flow chart and a narrative description for your manufacturing process. Again, phase one, it's generalized process flow. And you want to make sure that you include information on your sterilization process if you have a sterile product, or certainly aseptic processing if you have a sterile product that's produced by aseptic processing. And this is just an example of a typical oral tablet manufacturing process for early phase. Again, it's pretty general and provides basic flow of information. Next slide. Okay, this is go. Now we're going to excipients, and P4 is the section for excipients. And I wanted to talk about the specifications for your excipients. Hopefully you're using excipients that are included in the FDA inactive ingredient database. I provided two links here. One is frequently asked questions about the inactive ingredients in the database. And the other is the link so that you can search for those approved excipients that have been in approved drug products. It's really important to understand and review this information. I'm not going to go into more detail, but I expect this is going to be something that you might have some questions about. The suggestion always is, if possible, to avoid using novel excipients because additional safety testing equivalent to information needed for the drug substance is required for those for novel excipients, which includes tox testing and things like that. But for phase one, you provide a list of your excipients, not just your compendial ones, with any reference to quality standards. Next slide. Okay, so now we're going to talk about excipients of human or animal origin. Again, if you can get away from using excipients of human or animal origin, you're better off because the documentation you have to have is less, but you need a minimum of a BSc TSC certification. And then if there is, for example, a drug master file that includes information on the manufacturer of that excipient, then you want to be able to cross-reference that information in the drug master file. If you don't have information on the excipient, you must provide information regarding adventitious agents. You have to have the source, the specifications, the description of the testing performed, all sorts of viral data. And then that information is included in an appendix to the drug product section 3.2.A.2. If you have any questions about excipients of human or animal origin, let us know. Next slide. Novel excipients. And those are excipients, again, that have not been used for a drug product of the same type as yours and provided in the inactive ingredient database. So if you have a novel excipient, you have to provide full description of the characterization, manufacturer control, analytical procedures. You have to have tests and acceptance criteria. And again, the information that needs to be submitted for your novel excipient is almost the same as for a drug substance. So consider, if you're using a novel excipient, this information. Alternatively, if you have a reference, if you're using a novel excipient, but there's a reference to a drug master file from the manufacturer, then you want to be able to include that information. Next slide. Okay, so now we're going to talk about drug product specifications, and we're talking about drug product because we switched to P5. But for phase one, you provide test methods, again, and the acceptable limits for your critical quality attributes, but of course, specifically focusing on identity, strength, potency, quality, and purity. And again, the specifications that you provide can be broad specifications. They should be broad specifications because you have minimal batch history, and those specifications should be based on your batch history. And what you don't want to happen, if possible, is you don't want to have to be in a situation where for future batches or for phase two or phase three, you end up having to loosen your specification because that requires more justification. So include reasonable limits for your specifications. And my suggestion is to include that information in your pre-IND briefing document and get any feedback from the agency that you can, if they'll provide you feedback on the specific specifications. Next slide. And this is, again, this is just another example of a specification for a drug substance. It's for a round white tablet. The main thing I wanted you to see here is the related compounds, which are really the impurities. And we have them identified. You can do it by relative retention time, or like here, compound A, B, C, or D, where you noted that on your chromatogram, and you include a specific specification for that impurity. And again, if it's important, you want to make sure it's linked back to drug substance as appropriate. And you want to make sure that you tighten your specifications for your impurities as you go forward, if you can. And the other thing is oftentimes it's not realized, but for a drug substance or for a tablet, oftentimes the agency will want you to have an aerobic count and a yeast count. And that really is true depending on your study population. If you have compromised individuals taking the drug, you may want to include some additional information on microbial control. Next slide. Again, you have to include your analytical procedures for your drug product. Again, the high-level method descriptions and validation data is not required, but should be available by phase 2. And again, my comment about qualifying those methods as soon as you can, way before phase 2 is what I suggest. Next slide. Your container closure system for your drug product is important because that's what's going to do the clinical study. And again, it's the sum of the packaging components that contain and protect the drug product. So you provide summary information of the primary container closure. You can provide high-level diagrams. You provide reference to compendial standards. Again, there's a guidance document specifically relative to glass and polyethylene quality. And if you have or you're using an atypical delivery system, such as an MDI or an auto injector, you need to include specific information and diagrams for that specific device. And my suggestion is that phase 1, if you can get away with not using a device, don't. Next slide. Okay, so we have a stability summary and conclusion section. And again, it's the same thing. It's the critical quality attributes of the product. You provide a brief description of the stability study, the storage conditions, the test methods that you're using, if they're different than the methods provided for the drug product, the specific container closure, the testing time points, again, all of the information about manufacturing date and things like that. Then in this section, you provide a summary statement relative to the best storage conditions for the clinical trial material, and you provide information on an expiration date or a retest date. And I call it a retest date because you're going to add to the testing, and you're going to extend the time that you can use it in the clinical study as you collect your data. And one thing to know is there's no specific requirement for your stability study that you include in routine, intermediate, and accelerated conditions. ICH stability is for NDA or BLA. If you have the material to be able to do that at early phase, sure, include that information. But especially for biologics, if you don't have a lot of information, you don't have to have those 3 different conditions. And again, you monitor the stability for the length of time that from the date of manufacture until that clinical trial material is used in your clinical program. And so that's the time frame that your stability study needs to support. But again, I'm not suggesting that if you can get longer stability, it's important to go ahead and get an understanding of the stability of your product. But this is just so if you have a small amount of material, you understand information about stability. Next slide. Okay, the stability data again goes in a separate section. Again, you include the information on the container closure and everything like that. If your product is light sensitive or moisture sensitive, you need to include that and use that to support your selection of your container closure system. For your phase 1, routinely, it's a good idea to include at least 1 to 3 months of data on the clinical trial material batch that's at the proposed storage condition. And the reason you do that is because you need to commit to monitoring the stability, to support the stability of the clinical trial. Again, from the date of manufacture until that drug product is used in the stability study. And you have to commit to removing the clinical trial from the stability study if the stability goes outside your defined specifications. So that 1 to 3 month time frame gives you that time frame to be able to have enough stability to give yourself time to remove it from the clinical study if it's needed. Hopefully it won't be, but that's what you need to commit to. Next slide. Okay, so this is not in module 3 for the IND, but it is a part of the CMC information that was originally included in section 7 for the original IND, and it's also in the CFR. But what you do, there's a section in module 1, 112.13, it's called environmental analysis. And this is where you include the statement of categorical exclusion from environmental analysis for your IND. You must include that document. I've had INDs that I've had calls to tell me you didn't include an environmental analysis in your IND. So you must include that information. Next slide. And then the drug labeling information is, again, it's not in module 3, but it's included in 114.4.2. You provide information about a mock-up of your primary container label for your clinical trial material. One thing that's important to know is that the only specific information that you must have on that label is the caution statement. Caution, new drug, limited by federal or United States law to investigational use. That is the only thing that you have to have on there. You can put other information on that label as much as you have space, specific your lot number and your manufacturer usually. But the main thing you cannot have is you cannot put any statement that suggests that the investigational drug is safe or effective. Next slide. And one of the other things that I always include for early phase, for folks that are working on early phase that may not have lots of experience, is that there's no FDA requirement to include an expiration date on the primary container for your clinical trial material. That's really important because you'll get some investigational pharmacies that want to insist that you have the expiration date is included on the label. But FDA understands that it may be possible to extend the expiration date based on your stability data as it's generated. And not including the expiration date on the primary label makes it unnecessary to over-label or relabel clinical supplies as the expiration is extended. And this is especially important for clinical trial materials stored at refrigerator or freezer temperatures, because you can't defrost or allow those containers to reach room temperature, so you can reapply label. Next slide. So that's all I have to talk about for that. But now we're going to questions that were provided before the presentation. And I'm going to kind of address them in bulk. But here, questions about the pre-IND and what level of CMC information needs to be completed for a pre-IND. And again, what kind of manufacturing and analytical testing. I tell everybody that if you have a question about your CMC program, you want to specifically make sure that you've covered that information in the CMC information that you provide. But generally, what is provided is, again, a high-level flow chart, maybe a narrative of your manufacturing process for the drug substance and for the drug product. You provide a table of your testing for your specifications. You can include ranges for your specifications. And you may want to ask, a question: does the agency agree that the testing that you proposed is acceptable? Oftentimes they won't give you feedback on your specific specification, but you can try. And then let's see. Oh, oftentimes you want to include a bit of information about stability if you have it. Next slide. Okay, now we came to questions about the IND. And again, the specific differences between phase 1 and phase 2, 3, that's included on the slides. I hope you'll go back and review that information. Again, it's very high level, but at least gets you to thinking about what you need to do as your program progresses. And then again, what's required for CMC section of the IND, that's what I've provided for you. And then, do you need to produce a GMP drug substance or product to be used in the human study before the IND submission? Logistically, I think it's important to manufacture your drug substance batch before, because you have to provide information on the drug product. And so logistically, it's a good idea. You usually have some extended stability on that drug substance batch. And then as we discussed, for your drug product, you don't necessarily have to have the data from that batch in your IND submission. But again, I would have addressed this with the agency and gotten their agreement that you can provide the certificate of analysis for the batch before you start your clinical study. Okay, and then what are the differences in CMC requirements for IND versus phase 1 clinical? Your IND and phase 1 clinical should be the same. And a general understanding of the whole process, it's complex. There are, it's granular, very granular now, the way that the information needs to be put together. And so you really want to work with someone that has experience with CTD and especially eCTD. Next slide. Next slide. Oh, is there not another slide? Huh. Okay. There were a couple of other questions that came up. I'm sorry, I thought I had a slide for them but they were for other information that was more specific for a particular product. And what we're going to do is we have the email address for those that put those in, and I'm going to try to make sure that I provide you some feedback on those questions by using your email address. There were a couple of questions about devices. Again, I'm sending those questions over to our device folks. That's not me. You definitely don't want me to tell you what to do about devices. And then the other thing is there were a few questions about, or at least one question about what sort of tox. And again, I'm sending that over to the tox folks to give you some feedback for that. So that's all of the information so far and relative to the questions that came in early. So I'm now ready for questions that have come in during the presentation.
Carmella, there was one other question came in, says, what guidance is available for the development and qualification of a novel analytical method?
Oh, yeah. There are some good guidance documents. There's also good information on USP. Let me provide you with some guidance documents. I'll send you some references for that. But again, there's not gonna be specific information. You're gonna have to kind of use your best scientific judgment in developing the method. But again, that's a perfect opportunity or a perfect question to ask the agency at your pre-IND meeting. Does the agency agree with your method? Okay, anything else? Okay,
There was one comment on a transdermal delivery system and a drug device combination.
right. There's specific guidance for transdermal. There's a lot of information about information you need for your drug substance because you need further characterization of your drug substance. And then of course you need further information on your transdermal product. But there are a couple of guidances that we'll try to make sure we get you pointed in that direction. And then if you have any specific questions, about information or tests and specifications, ask us and we'll try to give you some feedback. And if we can't answer them, we'll try to get to the right person to answer them. Anything else? Okay. I hope this has been helpful. I know it's high level. We could talk for hours about drug substance and hours about drug product. And we are looking for what people want to hear in these Catalyze presentations. So if you think it's helpful or you think it would be helpful to have more detailed discussion about drug substance manufacturing or drug product or other things, please let us know, because our goal is to provide you with the information that you need to help you develop your products. I mean, that's what we're here for. Any other questions or comments? Okay, please do send us your questions. Go back when the presentation is up on the website, review it, have your postdocs or people that are working in your lab review that information and have them come back with some questions, because that will be the best way to provide you with information for your specific issue or something that's been a burning question you haven't gotten any feedback for yet. And again, it may be that we can't answer or I can't answer everything, but we'll do our best to make sure that we get to the information or to someone that can give you the best feedback on the information you've provided or that you've requested. Okay, Marissa, back to you.
Thank you so much, Carmella. I know that Mark is typing a few additional answers into the Q&A box, but this will be a great time to launch our evaluation poll, as I'm just going to go over a few upcoming events. So our next webinar will be a programmatic one. It's ahead of our next receipt date for product definition applications, which is due in June. So our webinar will go over application details as well as specific information about our Catalyze NOFOs. That will be on May 6th, and I am dropping our registration link in the chat right now. So that should have just gone through. Otherwise, I think that is all that I have. So as people are filling out our evaluation poll, please continue to feel free to drop additional questions in the Q&A. And as Carmella said, we'll be sure to follow up with you. There are several questions that were already submitted that we'll send email responses to as well.
I really enjoyed giving you the presentation. I hope it's helpful. Please do let me know how I can improve. I can't change my Southern accent, but I can provide different information if need be. Thank you.
Great. So we'll leave that evaluation poll up for just a few more minutes. And again, please do take the time to quickly register for our next webinar. There will be time during that opportunity for questions and answers as well. A great opportunity to engage with Catalyze leadership ahead of our application cycle. So thank you, everyone. And Patrick, I think we can close at the top of the hour. All right. Thank you, everyone, for joining. I think I've seen our responses kind of trickle off. So I think we'll go ahead and close. Thank you again for joining, and we'll see you next month.